Earth supports millions of macroscopic life forms. To understand them, we have to identify and categorize every single one. But human localization gets in the way. A specific apex predator is called a tiger in English, a huli in Kannada, and a bug in Bengali. This creates semantic ambiguity. When multiple researchers apply completely different local labels to the exact same biological entity, scientific data collection collides. To map evolutionary relationships accurately, biology requires a universal information architecture, one that ignores spoken language entirely. Relying on localized terminology fails at a global scale. A rigid universal data protocol is the only way to effectively systemize the biosphere. The technological solution developed to eradicate these naming errors is called Binomial Nomenclature. It is a strict two-part identification protocol. Every organism receives a generic name followed by a specific epithet. This is an illustration of the mango plant, demonstrating the protocol in action. Notice the universal formatting syntax required globally. The genus name is capitalized, the specific epithet is lowercase, and the text is italicized. Mangifera, indica. That binomial tag does not change regardless of the native language of the scientist observing the specimen. Assigning a binomial name permanently locks an organism to a specific, unique data point. This creates the foundation for all higher-level biological classification. The bedrock of this classification is the species. A species is a group of organisms with maximum genetic similarity, capable of interbreeding to produce viable offspring. The gray wolf, Canis lupus, and the domestic dog, Canis familiaris, are reproductively isolated. They are two distinct species. However, they share deep morphological similarities, which aggregate them into the first taxonomic level up, the genus node, in this case, Canis. We see this exact rule applied to apex predators. The lion, Panthera leo, and the tiger, Panthera tigris, share core anatomical frameworks. Those physical proportions group them together under the Panthera genus. The overarching system of categories used to group these organisms is called taxa. The genus serves as the crucial first step in data aggregation. Now consider the domestic cat, Felis catus. Its physical diverges enough from lions and tigers that it requires a separate genus entirely, Felis. To resolve this divergence, we introduce the next hierarchical level. The Panthera and Felis nodes converge into a single family folder, Felidae. This highlights a core taxonomic rule. As we ascend, required anatomical similarities decrease, but inclusiveness expands. Bringing back the domestic dog from the family Canidae, we test it against the cat family. Felidae and Canidae remain separate families, but converge at the higher order level, Carnivora. They share an evolutionary adaptation, flesh-eating. Higher taxa organize organisms by shared systemic functions and evolutionary survival strategies, rather than strictly identical physical forms. Scaling up to the class level introduces vastly different external morphologies, from bipedal humans to trunk-bearing elephants. Despite these external differences, both share a specific biological baseline, viviparity, or giving birth to live young. That reproductive trait forces these disparate organisms into the shared class Mammalia. For animals, the next tier up is the phylum, which groups distinct classes based on deep foundational structural architectures. To enter the phylum Chordata, an organism must meet specific criteria, specifically the presence of a notochord or a dorsal hollow neural system. Botany operates with a specific exception. Plant science utilizes the term division instead of phylum. Broad structural rules, like the presence of distinct floral identifiers, categorizes these press specimens into the division Magnoliophyta. The entire data structure is capped at the kingdom level. Grouping organisms into either Plantae or Animalia represents the highest macro tier, operating with the lowest possible barrier for shared characteristics. Visualized completely, the taxonomic hierarchy acts as an inverted framework. The top is wide and mathematically sparse, grouping massive evolutionary branches. The bottom is incredibly narrow and dense, containing millions of specific, localized data points. Moving downward through the hierarchy acts as an increase in morphological resolution. It filters out differences step by step until only exact matches remain. It is the intense sensation of rapidly descending through layers of biological complexity, ultimately arriving to find a single, highly specific origin point. This nested data structure allows scientists to systemize and retrieve relationships without data collisions across millions of species. We can test the architecture by running a comparative analysis of two completely distinct entities, a human, Homo sapiens, and a housefly, Musca domestica. Both organisms occupy the kingdom Animalia, but they diverge immediately at the next highest node due to their foundational builds. The fly lacks a dorsal neural system and relies entirely on an exoskeleton. Those factors separated immediately into the phylum Arthropoda. From there, the housefly makes a specific, isolated descent down the phylogenetic tree through the class Insecta to arrive at its precise binomial tag. This divergence highlights exactly where two evolutionary branches split millions of years ago. Taxonomy functions as a precise, hierarchical index of evolutionary and structural coordinates. We began with a planet swarming with chaotic, unintelligible biological noise. Systemization brings order to that chaos through rigid classification rules. Binomial nomenclature acts as the anchor, pinning every living entity to one undisputed data node. The hierarchical strata of systematics then wire all those individual nodes together into an accessible network. Systematic biology provides the structural framework for the living world, organizing an overwhelming biosphere into an elegant, queryable relational database.